In continuing the cell cycle, we will further now proceed to another process, that is the cytokinesis. Cytokinesis is the process by which cell divides its cytoplasm to produce two daughter cells. It is the last stage of cell cycle which completes the process of cell division. Cytokinesis follows every mitosis, but some of the cells, such as early Drosophila embryos, some mammalian hepatocytes, heart muscles cells, they undergo mitosis without the cytokinesis, and thus they acquire multiple nuclei. Cytokinesis in animal cells. There are two types of cells which we will deal in, that is the animal cells and the plant cells. During the cytokinesis in animal cells, cleavage, it or the furrows suddenly appears on the cell surface. Furrows rapidly deepens and spread around the cell and thus divide the cell into two parts, just as shown in these two pictures, where the furrows they are appearing and the cell is dividing into two parts. In this process, there is a contractile ring formed, and in interface cells, actin and myosin 2, that is the proteins, their filaments form a contractile network underlying the plasma membrane. As cells center mitosis, these arrays of actin and myosin they disassemble, that is, they undergo conformational changes, just as shown in this cartoon, that actin and myosin filaments of the contracting ring, these are the red one, they are appearing. The microtubules of the mitotic spindle, it determine the plane of animal cell division. In cytokinesis, it is important to ensure that division occurs at the right time and at the right place, just as shown in these the three current models of how the microtubules of the anaphase spindle generate signals that influence the positioning of the contractile ring. Just as if so, there is astral stimulation models, astral spindle stimulation model, that is the middle one, and then extreme left, the astral relaxation models, which divide the cell in the different planes. Cytokinesis in the plant cells, however, that occurs due to the fact that the plant cells, they have got a cell wall, and cell wall is very rigid. So unlike the animal cell, the cell wall can't squeeze. Instead, a structure called the cell plate, it forms across the middle of the cell, and the cell gradually splits into two parts. Thus, two new cell walls are formed. Just as shown in this figure, the extreme left, that is the G1, is a cell. Then in the telophase, the remains of interpolar spindle, its microtubules, they are formed, and Golgi cluster vesicles are appear. Then what happened that the early cell plate, it appears in the cytokinesis, and further G1, new cell wall, is formed. Now the phagomoplast, it guides the cytokinesis in the higher plants. The assembly of the cell plate begins in late anaphase and is guided by a structure called the phagomoplast, which contains microtubules derived from the mitotic spindle. Just as shown in this diagram, the arrow indicates the structure of the phagomoplate, and the cytokinesis in the plant cell during the telophase has been illustrated.